Hi there, welcome to this video, Respiration Stage 3 Krebs cycle. So let's go through a little bit of recap of the stages so far. There's separate videos for stages 1, 2 and 4, so watch those as well. So stage number 1 was glycolysis. Glycolysis happened in the cytoplasm of the cell, and glycolysis made a net of 2 ATP. And it also made, if we go back onto the videos, it also made, if we go back, I'm just trying to find it for you so you can visualize it. So, which one is it? Here's our final slide anyway. It made 2 reduced NADs as well. So the reduced NADs are just carrying the let's move this across. Just carrying the hydrogens to stage 4. Oh, they're stuck together. So first stage glycolysis, we've got a net of 2 ATPs. Making me carry everything individually. Net of 2 ATPs and 2 reduced NADs. Okay. Then the second video we looked at, stage 2, was the link reaction. The link reaction happens in the matrix of the mitochondria. It doesn't make any ATP, but it does make reduced NAD. So if we go back to our last slide of that one, here we go. So it made no ATP, but it made 4 reduced NADs. 4 reduced NADs. It's going to mess up the format again. Okay, too bad. So 4 reduced NAD. So we're now going to go on to the Krebs cycle, stage number 3, Krebs cycle. If you're trying to research it in various books or YouTube or whatever, sometimes it's called the citric acid cycle. Okay, so just in case you hear it called that, it's not something different, it's just another name for the same thing. Okay, so we'll go through this step by step and we'll look at how many ATPs and reduced NADs and the like it produces. It happens in the matrix, the same place as stage 2. Okay, so glycolysis was in the cytoplasm, link reaction, the matrix. The matrix, I should say, is the middle part of the mitochondria, in case you weren't sure of that. And the Krebs cycle is also in the matrix, the middle part of the mitochondria. And that's what we're going to do now. So we'll work out how many ATPs and reduced coenzymes it makes. So the first thing to understand is the numbers of carbons. So the molecule that's coming into this from the link reaction is acetyl coenzyme A. And acetyl coenzyme A, if you remember, is a molecule with 2 carbons. Okay, now it specifically says in your specification that you don't need to know all of the intermediate molecules. In your textbook, it does tell you the names of them, but you do not have to remember them all. It specifically says that in the spec. Okay, so what happens is that this 2 carbon molecule from acetyl coenzyme, or called acetyl coenzyme A, combines with a 4 carbon molecule. Right, so 4 plus 2, so this 2 carbon molecule is like coming in here, joining on, so the 4 carbons coming along here, the 2 carbons here, and at this point, look, they're joining. Yeah, they're reacting together to produce a new molecule. Now, if we've got 4 carbon and a 2 carbon, they're going to add together to make a 6 carbon molecule. Now, somehow, we need to finish off the Krebs cycle in a way that gets us back to 4 again. Okay, so in order to get to 4, we need to go via a 5 carbon, and then we can continue round and go to a 4 carbon. Now, just the same as the link reaction, the way we lose carbons, if you like, is by producing carbon dioxide. So a molecule of carbon dioxide is produced. carbon dioxide contains 1 carbon. So if a 6 carbon molecule loses a 1 carbon molecule, we're left with a 5 carbon. And the same thing happens again. If the 5 carbon molecule releases a molecule of carbon dioxide is made of 1 carbon, put carbons in there, and therefore we're then down to 4. And then it keeps going round and round and round. So 4 plus 2 makes 6. 6 loses 1 to make 5, loses 1 to make 4. 4 plus 2 makes 6. And it goes round and round and round and round. It doesn't produce any molecules that go on to stage 4 like the other stages have. The point of this is just that as it's going around and around, it's producing reduced coenzymes and ATP molecules as it goes around. Okay, so try and learn the numbers, etc., if you can. But if you can't, just learn the concept, learn the numbers going round, okay, and learn how many ATPs and reduced NADs it produces as an overall. Okay, if you can learn the whole cycle, well done, brilliant. But learn what you can. So after this carbon dioxide is lost here, we're going to produce a molecule of reduced NAD. So remember what happens. We start with NAD, and that picks up hydrogens. So hydrogens are given from this 6 carbon molecule, given to the NAD, and the NAD then becomes reduced NAD. Okay, remember all the time, oil rig, oxidation is loss, reduction is gain. So if it's gained a hydrogen, it's now called reduced NAD. Okay, then it releases another carbon dioxide, and in between there and the 4 carbon molecule that's made, a lot more is going to happen. So each time we produce reduced NAD, we should really, in full, draw it as this curved line like we have been doing. But because we've got so much to fit in, we're just going to simplify it. So I'm just going to draw it like this and say that we produce just there another reduced NAD. Sorry, that in. Not very good, that's better. Okay. Then we're going to go through a series of 4 carbon molecules because already it's down to 4, isn't it? As soon as it's lost that carbon dioxide, really. And one of the things that it's going to do is going to produce an ATP. So it would have made that from ADP and a phosphate, an inorganic phosphate, to make a molecule of ATP. We're then going to make another coenzyme, which we haven't come across yet. We've come across NAD as a way of carrying hydrogens, but now we're going to use an FAD to make a reduced FAD molecule. But it's the same thing, it's just going to carry hydrogens onto the last stage. And then we're going to make another reduced NAD. Okay. Then we've got our final version of the 4 carbon molecule, which we add to the 2 carbon to make a 6. 1 carbon dioxide is left, and an NAD gets reduced to give a 5. Then another carbon dioxide is released with a 1, which gives us a 4, but there's actually a series of lots of intermediates, 4 carbon molecules here before we get to this final one. And along the way, we make another reduced NAD from NAD, another ATP from ADP, a reduced FAD from FAD, and a reduced NAD from NAD. Okay, so we're making lots of useful molecules as we go through it. Now we can add a little bit more language so that we could put it into sentences with good descriptions. So each time we lose a carbon, remember, we can say that the molecule before has been decarboxylated. So here we could say that the 6 carbon molecule is decarboxylated when it loses the carbon dioxide. Here you could say the 5 carbon molecule is decarboxylated. Each time the NAD becomes reduced NAD, it's obvious that it becomes reduced because it's in the name, but the NAD has the molecule before has become oxidized. So the 6-carbon molecule becomes oxidized as it gives its hydrogen away. It also loses hydrogen, therefore it's dehydrogenated as well. Okay, so remember the word dehydrogenated means loses hydrogen. Decarboxylated loses carbon. Okay, so every time hydrogen's given, so hydrogen would need to be given to NAD to make reduced NAD. Hydrogen would need to be given to NAD to make reduced NAD. Hydrogen would need to be given to FAD to produce reduced FAD. And then hydrogen would need to be given to NAD to make reduced NAD. So each time that's what's happening. Here we're making a molecule of ATP from ADP. So ATP, if you remember, ADP adenosine diphosphate, and we're going to make ATP adenosine triphosphate. So here it's the molecule with the 2 phosphates. Here's the molecule with the 3 phosphates. Okay, so if we're adding a phosphate, we could say that ADP is being phosphorylated. Yeah, so ADP is phosphorylated to make ATP because we're adding a phosphate. All right. Now, in the textbook, if you want to look it up, it has got the names of some of the intermediates. You could learn the name of the 6-carbon, the 5-carbon, and the 4-carbon if you want to. That's up to you. So the 6-carbon molecule here, I'll put them on just in case anybody wants them. So the 6-carbon is called citric acid. Now that makes sense, doesn't it? Why they sometimes call it the citric acid cycle, because that's our 6-carbon molecule there is called citric acid. As we go round, we get to our 5-carbon molecule, then our 4-carbon molecule. I'm just looking, I don't know if it even tells you the names of the intermediate ones. The next one that's really famous is the 4-carbon, the final 4-carbon up here, which is called oxaloacetate. Okay, that's what the name of that 4-carbon one, just there. It doesn't even tell you the name of the other intermediates, so they can't ask you for them. That's good. Okay, so what's the Krebs cycle producing then? So the first thing when we're trying to add up what's being made is remember that this whole Krebs cycle is happening twice for each glucose, because each glucose made 2 pyruvate, so all of link reaction is times 2, yeah, and therefore all of this needs to be times 2 as well. So each Krebs cycle makes 1 ATP, but remember it's times 2, okay, because there's 2 pyruvates going into link and 2 acetyl coenzymes A going into their own Krebs cycles, okay. It makes 1, 2, 3 reduced NADs, I'll just put red NAD to save space, and 1 reduced FAD. All right, but remember they're times 2 again. So the total, if we go back to our summary that we had at the start where we were doing the additions of each one, here, oops, sorry, I didn't mean to go back off it. Let's change colour. So in terms of ATPs, it makes 1 ATP per cycle, but we're doing the cycle twice, so it's 2 ATP. It makes 3 reduced NADs per cycle, but we're doing it twice, so it's going to be 6 reduced NAD. And it makes 1 reduced FAD per cycle, but we're doing it twice, so it's going to make 2 reduced FAD. Okay, so that's our kind of running total. Now, don't forget the point of what we're doing. So the first purpose, if you like, of what we're doing is we're trying to produce ATP. So we've made a little bit of ATP there, we made a little bit of ATP there, but we're going to make lots of ATP in stage 4. The other purpose of what we've been doing in these early stages is giving our hydrogens to our coenzymes to carry to stage 4. So we got 2 reduced NADs there, 4 reduced NADs there, 6 reduced NADs there, and 2 reduced FADs just there. And all those pink ones are going to go on to stage 4 to oxidative phosphorylation to make much more ATP when we get there. Okay, and in respiration, most of the ATP that's made is in stage 4. It is in oxidative phosphorylation. That's where most of it happens. Okay, now if we calculate how much ATP is actually made, the calculation says that after the 4th stage, if I just do a little summary now, we call it oxidative phosphorylation. What happens in oxidative phosphorylation is that these reduced NADs are used to make ATP. Okay, now one reduced NAD can produce approximately 2.5 molecules of ATP when it gets to oxidative phosphorylation. So we've got 2 plus 4 plus 6 reduced NADs coming in, okay, times that by 2.5, because each one gives us approximately 2.5 ATP molecules as it goes through oxidative phosphorylation, okay, so 6 times 2 is 12, half of 6 is 3, so 15, okay, so from 6 reduced NADs, in stage 4, it can make 15 ATPs, and the reduced FADs, each FAD that gets reduced can produce about 1.5 molecules of ATP in oxidative phosphorylation. So those 2 reduced NADs that we've got coming in are times by 1.5, and therefore they will make about 3 ATP molecules. Okay, so these, oh, I didn't mean to do that. I meant to highlight. These reduced NADs that came from the earlier stages and the reduced, oh, that's a mistake. Look, I said FAD and wrote NAD. Apologies, let's correct that. FAD. So these reduced coenzymes that we got from the earlier stages in the stage 4 are going to make us loads of ATP. So if you then add together all the ATPs that we've made, I'll put these ones in brackets because we're not making them there, we just carried them there. If you add all those ATPs together, in total, it should make 32 ATP in the whole of respiration. Now, the reality is it doesn't actually make 32. It's probably more like 30. Some of the protons leak out. Some of the ATP is used up in active transport, moving pyruvate around and moving reduced NAD around. So more likely 30, but theoretically, it should make 32 ATP. So the point is you need to know that a little bit of ATP is made in glycolysis, a little bit of ATP is made in Krebs cycle. Glycolysis link and Krebs cycle build up these reduced coenzymes which carry hydrogens to the 4th stage, and then they're used in the 4th stage to make loads more ATP. And make sure you watch video 4 of how they then make all that ATP. Okay, and we'll finish there.